In this lecture, we're going to talk about nerve impulses, the electrical signals conducted along neurons, and also muscle cells can do this, and the study of which is called neurophysiology. So I wanted to start with this picture of a membrane protein in the membrane of a neuron. This is a amino acid structure folded into the membrane, and this is an ion gate, so it allows ions to pass through it and through the phospholipid bilayer. So I wanted to start with this because I want you to start thinking, start visualizing the neuron at this level, at the membrane level, because that's where it all happens. To understand nerve impulses, we have to zoom in on the membrane. So this gray box is, just a skeletal representation of the phospholipid bilayer, and then this is one of these channel proteins in the membrane, and they really play a key role in how it works. So in this slide, here's the phospholipid bilayer. Right now we're not seeing any membrane proteins, but so we can look on the outside of the cell, that's the light blue, or on the inside of the cell, the cytoplasm, and notice that without looking at all the text, just the picture, notice there's positive and negatively charged ions. These are atoms with charge, like sodium, calcium, magnesium, chloride ions, which have a negative charge, phosphate ions, bicarbonate ions, all kinds of ions, both in and outside of our cells. And in the big picture, there is no charge difference. In other words, our body fluids aren't electrically charged, where the inside and the outside have a different charge to them. However, if you look just at the membrane of, and this is true for all cells, if you look at just the membrane, you notice from this picture that the outside of it is more positively charged than the inside. So there is a charge difference. It's very local. It's a membrane phenomenon. You don't see it, like I said, if you look way outside and way inside the cell, but just right across the membrane, there is this charge difference. It's actually in all of your cells, not just neurons and muscle cells, but all of them. And you can measure it. They first did it in giant squid axons, where they took a microelectrode, put it right on the outside, another one on the inside, and measured a charge difference across the membrane. We call that the resting membrane potential. Okay, and they call it resting because this is when there's no nerve impulse. If you're talking about a neuron, there's no nerve impulse going on right now in the cell, but there is this charge difference. So it's an electrical difference across the membrane. Okay, now I'm actually going to spend a fair amount of time in this lecture talking about what's at the heart of the resting membrane potential, or what's actually allowing it to be there in the membrane, because it really is the setup for understanding what a nerve impulse is. So in other words, we have to start here. So let's look at what's behind this resting membrane potential, because in this picture it's hidden, because the answer is mainly the proteins in the membrane. So here we see another picture of a membrane. Let's say this is a neuron, and then notice these colored objects in there. These are membrane proteins. So there's two main types of membrane proteins that really together play a major role in this resting membrane potential, because it's not something that would exist on its own. Actually, your cells work very hard to create and maintain this resting membrane potential. So the resting is a misnomer. It's not like the cell is, taking a nap or something. It's actually very busy, because if you look over here at this purple protein in the membrane, this protein, we call it a pump. It is very busy. protein, and it's using ATP, so it takes a lot of energy. And if you recall from general biology, this if you have membrane proteins that run off ATP, sorry about that, then they're what they're doing is they're actively transporting solutes up their concentration gradients. If you want to move something across the membrane from where there's less of it to where there's more of it, you need active transport. Review that if you need to, because it's a key process in nerve impulses. Actively transporting solutes means moving them from lower to higher concentration. So that requires, sorry I keep doing that, that requires energy. So this pump, it's called the sodium-potassium pump because it's a double pump. It pumps sodium out and potassium in at the same time. And in fact, notice it's pumping unequally. So it's pumping 3 sodiums out for only 2 potassiums in. And if you just think for a moment about the charge that's being pumped out versus in, think of it this way. Sodium and potassium have an equal amount of positive charge. In other words, the amount of positive charge in a sodium ion is equivalent to the amount of positive charge in 1 potassium ion. So pumping 3 sodiums out for 2 potassiums in is another way to look at it. Is that this pump is pumping 3 positive charges out for only 2 in. So even though they're different ions, it's an unequal. It's pumping the charged ions unequally. So it's helping to generate this electrochemical gradient across the membrane. This charge difference across the membrane due to these chemicals, sodium and potassium. So the sodium-potassium pump is what we call the active force behind the resting membrane potential. There's a handout posted under Unit 3 that covers this. Okay, so that's the active force. There's also something called the passive force that involves these different proteins in the membrane called leakage channels, or, I'm so sorry I keep doing that, or sometimes I just call them channels. And these are quite different from the pump. They only allow diffusion, not active transport. Only diffusion. In other words, when they're open, which they are open in general, the ions can only diffuse from where there's more of them to where there's less. So in other words, down their concentration gradient. That's diffusion. So these are not involved in active transport. Notice they're not using any ATP. They only allow diffusion. So the sodium, notice how sodium's moving into the cell. That's because there's more sodium outside than inside. So it's diffusing in. It's actually opposite the pump. Sodium's being pumped out and then it's diffusing back in. That's why they call them leakage channels. And then potassium's doing the same thing, except it's in the opposite direction because there's more potassium in your cells than outside. So it also diffuses down its own gradient, which happens to be out of the cell. So at first it might seem like, why even have these leakage channels when they're counteracting the pump? But actually the resting membrane potential is dependent on really both of these factors, the active force and then the passive force behind the resting membrane potential. These leakage channels help because look at the picture closely. There's more potassium than sodium leakage channels. There's more. So this means that more potassium can diffuse out of the cell than sodium can diffuse in. And again, if you don't worry about whether it's sodium or potassium, just think about the charge. Like with the pump, more positive charge is leaving than coming into the cell. So you put the two together and it's the pump and the leakage channels that are responsible for the resting membrane potential. Now, typically it's given as negative 70 millivolts. They just decided to give the charge on the inside of the membrane when reporting the resting membrane potential, hence the negative sign. And then if you measure the difference across the membrane, like how much more positive charge is right outside than inside, it's about typically 70 millivolts, a thousandth of a volt. So we're not talking about high voltage here. But so negative 70 millivolts is the an average resting membrane potential. Okay, so this is what the neuron looks like when there's no nerve impulse going on. Now, what's going to happen during a nerve impulse? For example, an action potential. You heard that term when we studied skeletal muscle. That's what we called the nerve impulse coming down the axon. Action potential. That's what they call. So, what's going to happen during an action potential? Well, the simple definition is that the membrane potential is going to change. It's going to change from minus 70. It's not going to be minus 70 anymore. And that change in the resting membrane potential is going to move down the axon of the neuron. And when it does, the resting membrane potential is going to change. So here's the definition of action potentials and their cousins that we'll talk about in a minute, called graded potentials. They are due to changes in the membrane potential. So back to this picture. In this diagram of what's going on in the resting state, they're not showing you a third type of proteins that are in the membrane that are actually responsible for this change in the resting membrane potential that's going to happen. And these other proteins are called gates. There's sodium and potassium gates. Notice sodium and potassium are key players in nerve impulses. They're like the main actors. Anyway, there are these sodium and potassium gates. Here's what a potassium gate might be visualized as. There are also proteins in the membrane, but notice that they can be opened or closed. So unlike the channels, which are basically open, these can be in an open state or a closed state. And in the resting state, the gates are closed. That's why they didn't even show them to you in the previous diagram of the resting membrane potential, because they're there, but they're closed. So nothing's going through them, so they're not having any effect. During an action potential, they're going to open. So these are the two types of nerve impulses that are found in neurons: graded and action potentials. Graded potentials are what happen first in a neuron. They begin typically in the dendrites. They travel along the membrane to across the cell body to the axon. And as we'll see in a moment, their job, graded potentials, is to trigger action potentials in the axon. And then those travel down the axon to the very end, where this neuron will synapse with some other cell, like a muscle cell, like we saw with the neuromuscular junction. Or it could be a gland to cause it to secrete. Or it could be another neuron, because this is how your 99.9% of your neurons talk to each other, as they send action potentials to the next cell over, another neuron. So what's similar between graded and action potentials is that there are both changes in the resting membrane potential. So they both involve ion gates opening, these membrane proteins, gates. It's all about those gates. There are some differences, however. Before I get to that, though, some vocabulary here. Okay, when these ion gates open, the resting membrane potential becomes more negative than -70, as you see in this graph up here, then we call that hyperpolarization. And if the membrane becomes more positive than the resting membrane potential, even if it's just going from, say, -70 to -60, that's called depolarization. Okay, so those are terms you're going to need to know. And then if you hyperpolarize or depolarize, then eventually the membrane needs to come back to the resting state, the -70. That's called repolarization, returning to the resting membrane potential. So we're going to be using those terms. Hyperpolarize, more negative than -70. Depolarize, less negative than -70. And repolarize, return to -70. Okay, so what's different between graded potentials and action potentials? Well, one big difference is where you find them, as I just mentioned. Graded potentials travel along axons and across cell bodies to the beginning of the axon. You don't have graded potentials in axons. They travel to the axon because that's really their job, is to either trigger an action potential when they get there, we'll look at that in a moment, or if they're too weak, they don't. That's their job. Now, graded potentials can be strong or weak, so they do vary in amplitude. They can be strong or weak, and they as they travel along to the axon, they get weaker and weaker. That's what decremental means. So they're strong. They're the strongest when they're first initiated by a stimulus, and then they get weaker as they travel along. And then, as I mentioned, this last trait or function really is that they can generate action potentials. Action potentials, on the other hand, only travel along axons, nowhere else. They can go very, very far. Like some of them go all the way from your spinal cord to your foot. I mean, these are the longest cells in the whole body. So, these action potentials can go a long way. They are always the same. So there's no such thing as a strong or a weak action potential. They happen or they don't. It's like, remember we talked about muscle cell contraction, and I said that muscle cells either contract fully or they don't. It's an all or none phenomenon. Well, the same with an action potential. It either happens or it doesn't. And if it does happen, it's always the same strength. So remember I said graded potentials can be strong or weak, but not action potentials. So they can go long distances. They travel along axons, so I kind of visualize action potentials as like a train, traveling along a long railroad track. And they're triggered by graded potentials. So that's what causes them. And then they'll travel to a synapse, and you actually learned that happens with muscle cells. When they get to the end of the axon, they release neurotransmitter into a synaptic cleft. So we'll review that at the end. Okay, now this complicated looking picture, let's come back to that later. Let's look at the simpler version first. We're going to start with action potentials. Even though their graded potentials occur first, you actually need to know the action potential in more detail, and so I'm going to cover it first, and then we'll go back and talk about graded potentials. So it's kind of convenient to break down the action potential into sort of these different steps that you see happening here. So I'm going to show you some bigger pictures of them, but this is the overall figure. So in step 1, here we see it better. In the resting state, we already talked about this, right? This would be the resting membrane potential. But in this picture, they're showing you the gates, and notice they're closed. The green gate is the sodium gate. Oh, and by the way, for simplicity, I am simplifying this material somewhat. So you see there's 2 gates in the sodium gate, well, oh, they're calling it sodium channel, but actually gated channel is a better description, and just gates for short. Anyway, if either one of those gates are closed, we're going to say the sodium gate is closed. We are not going into the inactivation versus activation gates. We're not going to go into that much depth, but you're still going to get a lot out of what we're about to cover. Because basically, the bottom line is, for sodium gates to be open, both of those little doors, those little pieces of the protein, have to be opened. Again, try to visualize these proteins in the membrane. Remember troponin and tropomyosin on the thin filament, and how these were proteins that could move, or like the myosin head that could pivot up and down. Okay, well, this is just another example of proteins that have little movable parts. In this case, little gates that can open and close off the cavity inside where the sodium or potassium ions could go. And also, I digress here for a moment, but while we're looking at this picture, remember that proteins can be highly specific. So sodium goes through the sodium gates, but potassium doesn't, and same with the potassium gates. Potassium ions can go through there, but not sodium ions. So this allows for great specificity here. Also, I want to remind you that the only reason really any of this can work is because the sodium and potassium ions themselves cannot go through the lipid bilayer. They are not soluble in it. If they were, who would need the gates? They would just zip right down their gradients. But they can't. So this allows for controlled movement of ions across the membrane, and that's what nerve impulses are. So these gates are key. Really, you could say that the action potential is due primarily to the opening and closing of these gates. So, in the resting state, those gates are closed. Now, here's what happens in an action potential. The first thing that happens is the sodium gates open first. They open when a graded potential arrives at the the beginning of the axon. You have sodium gates in there, and potassium gates. And the potassium gates, the sodium gates open when the graded potential arrives. This graded potential is bringing positive charge, so the fancy way to say it is that the membrane depolarizes. See that over here? Oh, I'm so sorry. That means a positive charge arrives at the beginning of the axon and opens the sodium gates. So these gates whether the sodium gate is opened or closed is dependent on the membrane potential. This is key to understanding how this works. At -70, the sodium gates are closed. But if you bring enough positive charge along the membrane from a graded potential, if there's enough of it that arrives, it can open those gates. So they're electrically controlled. This kind of gate is described as a voltage-dependent gate. Okay, and don't let that, make it more complicated than it is. It just means the gate is open or closed depending on the membrane potential. At -70, the gate is closed. You got to raise you got to depolarize the cell about 20 or 25 millivolts to make it like -55. Not sure, I just did the math right there. You have to depolarize the cell to open the gates. And when they do open, sodium floods into the cell because there's a gradient for it to move in. And so look at what is happening to the membrane there. It's getting more and more positive as sodium ions rush in. This is what we call the depolarizing phase of the action potential, when the sodium gates open. Now, only the ones at the beginning of the axon open, not the ones way down at the other end. However, when these first group of gates open and positive charge comes in, it diffuses next door and opens the next group of sodium gates. And then that lets more sodium in, and then that opens the next group. So the action potential literally does move down the axon as these sodium gates flip open like dominoes. And by the time they're flipping open at the end of the axon, the ones at the beginning have already closed, because I think it's best for simplicity to think of these gates as they open briefly and then they shut. So it's not like they stay open. They're like a swing gate with a spring. You open it, some sodium rushes in, and they swing shut. Okay, so that's like the first thing that happens in the action potential. In the second phase, guess what? You'll never guess. The potassium gates are going to open. And they pretty much open right after the sodium gates close. So when they open, as you see in this picture, and notice the sodium gates are closed now, so no more sodium's rushing in. Now, when the potassium gates open, potassium leaves. Because, why? Because there's way more potassium in your cells than outside of your cells. So potassium's diffusing out. What is that doing to the membrane potential? Well, if that's the only thing that's changing, I mean, there's still the pump and the leakage channels. They're always doing their thing. But if this is the thing that's changing, the potassium gates opening, then the membrane is going to start repolarizing. It's going to start becoming more negative inside again. This is called the repolarizing phase. Here it is in red. And then so the potassium gate's also a voltage dependent gate, but it just opens later. So, it's the sodium gate opens first, you get the depolarizing phase, that's the green part, and then the potassium gates open, you get the repolarizing phase, that's the red part. And this is a little bit of oversimplification, but basically then those potassium gates shut and it's over, and the membrane returns to -70. And that change you see there in the graph, the green and the red, that is the action potential. And that is going to spread down the axon like a wave. It's a wave of charge. See the y-axis? I'm afraid to use my cursor. It's moving the slide. In millivolts. So there's this, voltage, this charge, wave of charge moving down the membrane. That's the action potential. Now in this, okay, so here's at the end. Both the gates are closed again, and it's over. So notice the membrane potential is at -70 again. So, that's basically it. Just try to summarize it quickly. You could say that an action potential is when the graded potential arrives and opens sodium gates. Sodium rushes in and depolarizes the membrane. They close, then the potassium gates open, and you get repolarization. Then they close, and it's over. So it's all about these gates, and it's key that you understand diffusion and active transport. So please review it if you need to. It's the only way to truly understand this, is to understand why does sodium come in when you open the sodium gates? Why does potassium leave? It's about diffusion. Okay, now this we saw this briefly before. This graph shows a little more detail. It shows, notice over here where the stimulus is shown on the graph. This is annoying, I know, I'm sorry. That's when the graded potential arrives at the beginning of the axon. And notice that there's enough positive charge to depolarize the membrane to -55. So that -55 is really the key. They call it the threshold stimulus. That's what you have to depolarize the membrane to before the sodium gates will open. So the sodium gates don't actually open until you see the green part of the graph. That's when they open. So the stimulus, again, is the graded potential bringing positive charge to the axon, opening those sodium gates at -55 millivolts. What happens if the graded potential arrives and it can only boost the membrane up to, say, -65 millivolts? Well, nothing will happen is the answer. That's not enough to open those sodium gates. You know, they have a minimum requirement to open. Now, what happens if it went up to -40? Well, it'll open. It's like pulling the trigger of a gun. You just need to pull it hard enough. And if you pull it harder, it's not like the bullet's going to go faster or something. So it's, -55, you can think of it as the minimum amount of positive charge that has to arrive. And then you see the green depolarizing phase when the sodium gates are open. Again, the red repolarizing phase when those close and the potassium gates are open. And now in this graph, they show you how the potassium gates stay open even past getting back to -70. So there's a brief they call it the hyperpolarizing phase. But then those potassium gates close and it pretty quickly goes back up to -70. So this is a more detailed picture of an action potential. Why does having a myelin sheath, we talked about that in the previous lecture, why does having a myelin sheath make the action potential go faster? Because if you recall, not all axons have a myelin sheath. So I guess the short answer, which is the one I'm going to give you, the very short answer is that if you have no myelin sheath, then to keep the action potential going from the beginning to the end of the axon, you have to have many gates all along the membrane pretty close together. So, it takes a certain amount of time for all those little sodium gates to flip open like a bunch of dominoes. And then the same with the potassium gates. So it takes, of course, we're talking still milliseconds, thousandths of a second. It's still pretty fast, but if you have an unmyelinated axon, you need lots of sodium and potassium gates, is my point. If you myelinate the axon, then that's what these little boxes here are. These are Schwann cells here. Then you only have to have sodium and potassium gates in between the Schwann cells in the so-called nodes of Rambier. And you only need gates there and at the beginning of the axon, what they call the trigger zone. So because the myelin sheath insulates the axon and nothing can go in and out of the membrane where it's myelinated. So it's like you open the gates at the beginning or trigger zone, or sometimes called the hillock. Sodium will move in, it'll zip to the first node of Rambier, and open sodium gates there, and so on. So you need fewer ion gates. They're spread out further with these insulated regions of the axon in between. And they call it saltatory conduction because it seems like the action potential is jumping along the axon because you only have gates at intervals. And then, so it's faster. So the two main functions of the myelin sheath are to increase the rate of the action potential, not the strength, but how fast it moves. And also, if you damage this axon, they can help repair it. That's their other function. Okay, this is just another picture showing the myelin sheath. And remember that in the CNS, instead of Schwann cells, it's oligodendrocytes that are used. Again, that's in the previous lecture. Okay, so that's action potentials. Now, what about graded potentials? Well, first of all, we're not going to go into as much detail about them. But you do need to know the following. They're also due to ion gates. Typically, the ion gates involved are here at the end of the axons. I mean, there's variation, but that's typically where you find them. They are not located all along the dendrites and across the cell body. But they are at the ends of the dendrites. And so graded potentials are also due to ion gates opening. But they're not the same ion gates as you find in the axon. Some of them are sodium gates, some are potassium gates, but there also might be calcium gates, chloride gates. So there's quite a bit of variation. We're not going to worry about that in this course, in this class. but you do need to know that there's some kind of ion gates there. Whatever they are, there's ion gates, and that when they open, basically two things could happen. You could get an increase in positive charge or depolarization in the membrane that will travel to the beginning of the axon, or you could get positive charge moving to the beginning of the axon. In other words, you could get waves of depolarization or hyperpolarization. And that's important that both could happen, because to get an action potential, you must have a wave of depolarization, positive charge. Because remember I said to open those sodium gates, the membrane has to depolarize from -70 to -55. So only the arrival of positive charge is going to do that. So depolarizing graded potentials trigger action potentials if they're strong enough. Hyperpolarizing action potentials, when they get to the axon, beginning of the axon, they definitely do not cause action potentials. In fact, they inhibit them because they drive the membrane further away from the threshold stimulus of -55. What if you hyperpolarize it to -90? Now the membrane's even further from an action potential being triggered. And also you need to understand the difference between these two vocabulary terms. In sensory neurons, we call graded potentials receptor potentials or generator potentials. In motor and interneurons, they're called postsynaptic potentials. And they come in two types, E and I PSPs, excitatory and inhibitory. So I'm going to describe these in a moment. So keep these two terms in mind. We're talking about graded potentials here. That's not different between the different types of neurons, but they do use different terms. Receptor potentials, we only use that term with sensory neurons. PSPs for short, because it's such a long term, in motor and interneurons. So let's talk about the ions, the ion gates rather, that are responsible for graded potentials. First of all, they do have to open for ions to move across to change the membrane potential. So that's the same. The big difference for graded versus action potentials is in what exactly opens the gate. In the axon, it was the voltage across the membrane. Remember I said they were called voltage dependent gates. So that little piece of protein that served as the gate, the arm, it would only go up to open the gate if the membrane potential changed from -70 enough. For motor and interneurons, they do not use voltage dependent gates to open the gate. They use other stimuli. So for example, in some cases they use a mechanical stimulus where actual physical deformation of the gate would open it. So this would be true, for example, of your touch sensory neurons in your skin, or pressure receptors in your skin. They have a mechanical stimulus that opens them. There's also such a thing as, and this is very common, ion gates shown here in the middle that are open when a chemical binds to them. So they're called ligand, or some people say ligand, controlled gates. So that just means they're chemically controlled. So not by a voltage change in the membrane, not by mechanical force, pressure, but by a chemical binding to the gate. Oh, so sorry, I don't know why I just did that. So if you look closely at this picture in the middle, it's a little hard to see, especially when I keep moving it. Notice acetylcholine is binding to that gate, and that's what's opening it. This is actually what happened at the neuromuscular junction in skeletal muscle. Remember how you learned that acetylcholine gets released in the synaptic cleft, and then it binds to receptors on the muscle cell and causes a muscle action potential? Well, this is how. By acetylcholine binding to the gate opens it. And sometimes that causes positive charge to come in, like at the neuromuscular junction, but then there's also such a thing as hyperpolarizing graded potentials, where we have negative charge coming in. So, the main takeaway here is that the ion gates in graded potentials are controlled differently, either by chemicals or mechanical force. So here we see ligand-controlled gates for graded potentials, and then in green you see the voltage-dependent sodium and potassium gates. Again, that just means what exactly is opening those doors. Is it a chemical? Is it pressure, like mechanical force, or is it a change in the membrane potential? So for graded potentials in motor and interneurons, if it's a positive charge that is a depolarizing current that happens, then that can trigger action potentials. That's why they call it an EPSP, E for excitatory. If it's a wave of negative charge or hyperpolarizing signal, when that gets to the beginning of the axon, that's going to inhibit action potentials. And again, I know I'm repeating over and over, but why? Because the sodium gates need a depolarizing signal that will depolarize the membrane by several millivolts, from usually -70 to -55. That's depolarization. So E for excitatory. They excite action potentials. Not always. If they're too weak, they won't, but you know, that's their job if they're strong enough. IPSPs inhibit action potentials. Alright, I'm going to skip that picture. So this is just reminding you what depolarization is. The resting membrane potential went up in the positive direction, and then so like with acetylcholine binding to ion gates, that let in positive charge. And then you can have negative charge, you know, the inside the membrane becoming more negative. That's a hyperpolarizing graded potential. So there are ion gates that can also allow that to happen. So if you notice in this picture, I'm afraid to use the cursor, the chloride ions are coming in through that green gate that has a negative charge. So that's a hyperpolarizing ion gate. So in the big picture, whether or not an action potential occurs in an axon of a neuron really is not decided by just one graded potential somewhere. Motor and interneurons, remember they're called multipolar, they have many dendrites, and each one of those dendrites can synapse with a different neuron. So here you see a bunch of neurons out here. We call those presynaptic neurons because they're before the synapse. Here's the synapse in between the end of the axon of the presynaptic neuron and the dendrites of this would be the postsynaptic neuron in this case. So just like at the neuromuscular junction, you send an action potential down the presynaptic neuron, some kind of neurotransmitter gets released, it diffuses across the synaptic cleft, this white space, binds to receptors on the neuron dendrite on the other side, and then whether that gives you an E or an IPSP depends on what kind of ion gates are there. Are they going to let in positive charge or negative charge? Or they might let one or the other out. But the point is you know, is it going to cause a depolarizing or a hyperpolarizing signal? And notice in this picture, because you know, this shows you how complicated it really is in terms of, is this neuron right here going to trigger an action potential or not? Well, it can be, if you send action potentials down a bunch of different presynaptic neurons at the same time, you can have both E and IPSPs at the same time traveling down different dendrites, all going to the trigger zone of the axon. And when they get there, they summate. Summate means they add and subtract from each other. So, or you could think of it as voting. The EPSPs are voting, yes, we want an action potential. The IPSPs are like, no, we don't want an action potential. And when they get there, they add and subtract. You know, how much more positive charge is arriving than negative charge, or vice versa. So they summate, you kind of add and subtract, and you see what you get. If there's enough positive charge to depolarize the membrane there in the trigger zone to -55, then, like pulling the trigger of a gun, there is no stopping that action potential. Those sodium gates will swing open, sodium will rush in, and there's no going back after that. Everything is like flicking the first domino. The next one's going to fall, and then the next one, then the next one. Those gates are going to flip open from the beginning of the axon to the end. First sodium, then potassium, and you're going to get an action potential. But, it's not like one neuron synapses with one other neuron, and then that synapses with one other neuron. You see, it gets very complicated. Lots of input to this postsynaptic neuron in terms of whether or not it fires an action potential. Okay, one more slide. There's one more after this. Okay, this should be very familiar to you, I hope. This looks like the neuromuscular junction, doesn't it? You have the axon, the synaptic cleft, and then on the other side we looked at a muscle cell with receptors shown in green here. Okay, well in this picture it's the same setup, except on the other side of the synapse, instead of a muscle cell, there's a postsynaptic neuron, another neuron, probably the dendrites of it. Okay, this is a chemical synapse between two neurons, but very similar, once again, to the neuromuscular junction. You've got vesicles of neurotransmitter. It might not be acetylcholine, it might be serotonin or dopamine or glycine or some other neurotransmitter. But, it's something, some neurotransmitter. And then on the other side of the synapse, you have receptors for that neurotransmitter. Okay, and when are you going to release the neurotransmitter? When a nerve impulse or action potential comes along. So you got to send an action potential down this neuron. That's what triggers the release of the neurotransmitter. It diffuses across the cleft and binds to receptors on the other side. This should all seem familiar. And then this is a ligand-gated channel, or a chemically controlled gate. So the neurotransmitter binds to it and the gate opens. And then depending on what kind of gate it is, you're going to get depolarizing or hyperpolarizing, positive or negative charge. If it's depolarization's caused, then we call this an excitatory chemical synapse. If it causes an IPSP, it's an inhibitory synapse, if it lets in negative charge. So they would look similar. It's just, what kind of gate is this? What kind of ion gate? What kind of ions are coming in or out of the cell? Is it becoming more positive inside or more negative? Because that's what's going to spread to the beginning of the axon. So just a couple more things, and we'll wrap this up, that I want to say. This is a complicated topic, but try not to be overwhelmed by it. Try again to zero in and focus on those membrane proteins, the pumps, the leakage channels, and for the graded and action potentials, the gates, the ion gates. So now this might sound confusing at first, but I'll say it twice. In a given neuron, in a single neuron, the graded potentials trigger action potentials in the same neuron. So this nerve impulse here coming down this axon was caused by a graded potential in that same neuron. So graded potentials cause action potentials in the same neuron. Action potentials, on the other hand, travel to the end of the axon, release neurotransmitter, bind to ion gates on the other side, and trigger graded potentials in the postsynaptic neuron they synapse with. So action potentials cause graded potentials in the postsynaptic neurons that they synapse with. So let me say that again, and you can think about it. Graded potentials cause action potentials within the same neuron, and then action potentials from one neuron, the presynaptic neuron, cause graded potentials in the postsynaptic neuron that they synapse with. So that's how you get these neural networks where graded potentials trigger action potentials, and that those in turn cause graded potentials in other cells, and so on. So for motor and interneurons, the signal that causes the graded potentials is always a neurotransmitter. EPSPs and IPSPs are always caused by neurotransmitters. So they have a chemically controlled gates, ligand gated channels. For sensory neurons, it's going to be different. They are not triggered by neurotransmitters. Their gates are opened by environmental stimuli. Just think about what we talked about in the previous lecture. What do sensory neurons do? They monitor our internal and external environment. So their gates are going to be tied to environmental stimuli. Light opens sensory neuron ion gates in your eye. Pressure, touch, temperature from your skin. Chemicals in food or in the air for smell. Pressure in your blood vessels internally. Concentration of oxygen in your blood. So environmental, both external and internal stimuli, that's what causes receptor potentials. So there's still ion gates, but they're going to be either mechanically controlled gates, if they're pressure or touch receptors, for instance, or there's plenty of chemical gates, for taste, smell, and internal monitoring of body fluids. Okay, one last thing I promise. I don't think I have a picture, of it. Okay, there is such a thing as an electrical synapse where the two neurons, you don't need neurotransmitter because the two neurons are connected by something called a gap junction that electrically couples them. They're rare in the nervous system, quite rare, but you do need to know what they are. They're on your objective sheet. They're called electrical synapse. So do look at the picture in the book of those. Now they are common between your cardiac muscle cells and your heart, so you'll talk about those in 213 when you study the heart. Okay, so well, that was a lot of information, and let's see if I left anything out. A repair of axons, just to remind me to tell you that don't worry about going into great detail about Wallerian degeneration, etc. Just know that if you damage an axon, like it gets crushed or severed, then maybe, if you're lucky, your Schwann cells or your oligodendrocytes will be able to repair it. But we're not going to be going into the steps of it. So, this is a very long video, but it covers most of what you need to know for neurophysiology. Feel free to email me if you have any questions. And in our next lecture, we'll talk about the brain. Those are the topics for your next lecture test. The introduction of the nervous system that I already posted, this neurophysiology, and the brain.